name is Matthew Hudson. I'm an undergraduate researcher in the WVU Vaccine Development Center, and my research is focused on understanding relationships between cell size and colony size in Heme Oxygenase Mutated Pseudomonas aeruginosa. Pseudomonas aeruginosa is a gram-negative bacteria that is responsible for a great deal of hospital-acquired pneumonia and urinary tract infections. The Centers for Disease Control and Prevention identified gram-negative bacilli, including Pseudomonas aeruginosa, as paramount threats to global health due to the high rates of antibiotic resistance. While no vaccine currently exists for Pseudomonas aeruginosa and resistance continues to evolve, the need for a vaccine only increases. Our laboratory has an overarching goal of developing vaccines that target the bacterial acquisition of iron, which we know is a nutrient necessary for cellular and metabolic processes. To further study this, we partnered with collaborators to disrupt the heme acquisition system via deletion of the heme oxygenase gene. This mutant strain did not infect as well as the parental strain and resulted in the formation of small colony variants. Using scanning electron microscopy, we set out to determine if the genetic mutations to the heme oxygenase gene that affected colony size also affected cell size. The bacteria was cultivated on Pseudomonas isolation agar, and images were taken to determine colony size. The Pseudomonas aeruginosa was then used to coat the filter paper for scanning electron microscopy. The imaging was completed here at WVU. Due to the ongoing COVID-19 pandemic, in-person research was not possible. However, continued study of the topic was feasible through the use of the ImageJ software, which allowed for the remote analysis of colony diameter in the parental strain and mutant strain, as well as cell length and width in each of the 5 strains, as seen in Figure 1. Images that were captured here at WVU resembled that of Figure 3, which was a specific image from the QM strain. Statistical differences in cell width and length were analyzed using an ANOVA with multiple comparisons. While the results in Table 1 might seem insignificant at first glance, there are some statistical differences between the strains. Figure 5 highlights the genealogy of strains under study. The original Pseudomonas strain, PA01, was mutated, and the resulting strain is referred to as QM. Mice were infected with QM, and after dissection, 2 phenotypes were observed: a small colony variant, which was strain 13, and a large colony variant, which was strain 11. Strain 13 could then be reverted to the phenotypes of QM and 11, and the resulting strain became known as strain 28. We predicted that QM, 11, and 28 would have similar phenotypes to strain 13, but differ in some manner. Preliminary results indicate that our hypothesis, however, was not supported, and no obvious relationships between colony size and cell size were observed. Strain 13 was significantly shorter than all strains besides QM. However, the size relationships between strain 13 and remaining strains were not consistent with regards to the width. Further research is necessary to understand the causes behind such unexpected differences in the data. Determining if there were any contaminations or mutations among the strains throughout the duration of the study can be eliminated through the use of genetic sequencing. Since Pseudomonas aeruginosa is known to form biofilms that could adversely affect growth, it is worth studying to determine potential confounding effects. It will prove beneficial to plot growth curves between the strains to ensure the differences in growth cannot be attributed to incomplete growth. Further, the cellular metabolism of each bacterial strain is unknown, but could also account for size differences. As our understanding of Pseudomonas aeruginosa evolves, we hope to use our knowledge to create an effective vaccine for humans. Studying the effects of mutations to the iron acquisition system is yet another way of furthering our understanding in hopes of creating such vaccine. I would like to thank my fellow lab members for their expertise and guidance, as well as the National Institutes of Health, Cystic Fibrosis Foundation, West Virginia Higher Education Policy Commission, and WVU for funding this project. Thank you for listening.